All right, so this first learning target video is going to cover actually 10 learning targets, and I know that's a lot. Normally it's usually like 4 or 5 at the most, but all 10 of those have to do with the light-independent reactions, or also known as the dark reactions or the Calvin cycle, and so I feel like it's easier just to do it all in one video. So this might be a little bit longer. I hope not. I'm gonna try to keep it short, but just to give you a heads up that it might be a little bit longer. All right. So what we are looking at is the light-dependent reaction, and I'll try to go through and mention the learning targets as we go. Let me zoom out a little bit. I'm zoomed in for something else. All right, so the first is I can explain the purpose of the light-independent reactions. So let's review really quick the light-dependent reactions, which is not what we are talking about right now. We're talking about the light-independent, but I just want to talk really quickly. The light-independent reactions are going to produce a couple of things. They're going to produce oxygen gas, which is a waste product, so that doesn't really count. And then they're going to produce ATP and NADPH, okay? And that's going to be through movement of electrons, hydrogen ion gradient, ATP synthase, you know, electron transport chain, chemiosmosis, all that good stuff. So these are the two that we're really excited about for the light-independent reactions. And so the purpose of the light-dependent reactions was to collect the sun's energy and put it into these two guys. The purpose of the light-independent reaction is to take the energy of ATP and NADPH, which if you'll remember, came from the sunshine. So ultimately we're talking about the sun's energy, but we've converted it to ATP and NADPH, and to join or to take that energy out of those and put it into a 3-carbon, which will eventually become glucose. Because the whole idea of photosynthesis, these two reactions, is to take the light energy, the sunlight energy, and convert it into glucose. And so step 1, we convert it to ATP and NADPH, and then in step 2, we convert these to glucose. All right, I can list the reactants of the light-independent reaction. So the reactants are going to be 9 ATP, 6 NADPH, and 3 carbon dioxides. Okay, then the next one says I can list the products of the light-independent reactions. That's going to be 9 ADP, because we're going to break all of those ATPs down, 6 NADP plus, because we're going to oxidize all of those, remove their electrons, and then a 3-carbon, which we call glyceraldehyde 3-phosphate, or you might see G3P. You might see neither of those. You might just see a 3-carbon. And so we're going to have to do this process twice to get 2 of these to join them together to make that 6-carbon glucose. All right, I can list the 3 steps and phases of the light-independent reactions. At this point, I feel like we need to stop and draw, okay? And then we'll go back and label some things. So what we're going to do is we are going to start with 3 5-carbon molecules called ribulose bisphosphate. Okay, and I like to draw, as you've probably seen in my other videos, I like to draw these out, the carbons, just for those of you that are more visual, you can kind of see what's going on. So these are my 3 5-carbon molecules, okay? We are going to join those 3 5-carbons with 3 carbon dioxides. I don't know what I was trying to write there, but CO2. Okay, CO2 has 1 carbon. So this is going to be 1 CO2, 2 CO2, 3 CO2. So what we're doing is we're taking each of these 5 carbons and we're adding a CO2 to make a 6-carbon, okay? So we're going to make 3 6-carbon molecules. And that step is called carbon fixation. Let me write that in a different color so it kind of stands out. Okay, so that step is carbon fixation. And to fix, when we talk about fixing something, it's not like fixing something that's broken. It's more like fixing something, sticking it to it, attaching it for a permanent. So when we add a small molecule to a larger one to make an even bigger one, that's called fixing it in science. So we are fixing this carbon dioxide by adding it to a 5-carbon to make a 6-carbon. Okay, so carbon fixation is step 1. This 6-carbon is super unstable. Okay, almost immediately after it forms, it breaks into, let me draw it out here so you can see what I'm talking about. So here's my 6 carbons. Okay, but it's unstable, and almost immediately those break in half. And so we go from having 3 6 carbons to 1, 2, 3, 4, 5, 6 3 carbons. Okay, still 18 carbons, they're just in a different form. Okay, so let me draw those out. Now this actually happens in 2 steps, and I'm telling you that just to be transparent, but I'm going to show it as 1. So generally what we would do is we would show that break. We would have that the 6 3 carbons here, and then we would show this next step, but I'm just going to show it all as 1 step. You have 6 ATP, which are broken down into ADP. So that's going to break off that 3rd phosphate. It's going to release energy. That energy is going to be dumped into these molecules. We're also going to oxidize, whoops, I forgot the 6 NADPHs, which means we'll remove their electrons. That's going to turn them back into NADP+. Okay, and those electrons, which have all of their energy that they got from photosystem 1 and the sunlight that was absorbed by photosystem 1, if you don't remember that, go back and review the light-dependent reactions. Those electrons are going to be full of energy. So when they get transferred from the NADPH to these 3 carbons, they're going to take all of that energy with them. And so this is step 2, and it is reduction, because we are reducing, and that's what I didn't put up here. This is oxidized. This is being reduced. Okay, so the molecules are getting all this energy. I call this the energy dump stage, because we are dumping all of the energy from these ATPs and these NADPHs into this molecule. Because again, that's the goal. The goal is to get that energy into what's going to become glucose. All right. One of those 3 carbons jumps ship. It says peace out, see you later. So there's that. If you had 6 and you lost 1, you are now down to 5 3 carbons. Okay, so we're going to draw that like that. Okay, and then the last step, if you count those up, that's 5 3 carbons, that's 15 carbons. You are going to rearrange those into 3 5 carbons. Still 15, they're just rearranged, and that is going to take a little bit more ATP. So we're going to burn 3 more ATP doing that rearranging, okay? And so this last step, this 3rd step, is the regeneration of RuBP. So we've got to regenerate or remake these 5 carbons as RuBP so that we can keep going. All right, so it says I can list the 3 steps or phases of the light-independent reactions: carbon fixation, reduction, regeneration of RuBP. I can explain the concept of carbon fixation. Okay, again, that's just taking a smaller carbon molecule and adding it to a larger one to make an even bigger one. I can explain how during the reduction phase, so down here, large amounts of energy are passed from ATP and NADPH to the carbon-based molecules. Again, we break off that 3rd phosphate, the cell captures that energy and gives it to these. Here, that energy is moving with electrons. So we remove the electrons from here and we dump them onto this guy, reducing it, and that takes the energy with it. The next one says I can explain how carbon atoms are rearranged to form RuBP. So we have these 3 carbons. We're just going to rearrange them. We're going to break some bonds and we're going to reform them. Probably going to kind of break off these bottom 2 and add them to these top 3 to get 5, 5, and 5. And that's all you need to know. You don't need to know specifically which ones get broken and how. You just need to know that we had 5 3s and those 15 carbons got rearranged into 3 5s, which is still 15 carbons. I can explain why RuBP needs to be regenerated. Okay, well, it's a cycle. And so with a cycle, you always need to end with what you started with. So in order for us to start with RuBP, we have to end with RuBP. Otherwise, the cycle wouldn't happen, or we would have to do some other step over here somewhere to make this RuBP so that we could do this reaction. So it's just, since we've got the right number of carbons, it's just easier to do it right there. I can identify the location of the light-dependent reactions. Oh, Ms. Pirro, that's a good question, and you didn't tell us that. I sure didn't. The location is the stroma. Okay, so location is the stroma, which is good because if you go back and look at the light-dependent reactions, these 2 products are produced on the outside of the thylakoid, aka inside the stroma, or in the, I shouldn't say inside, the stroma is like a liquidy, jelly-like stuff. So they're already in the stroma where they need to be. And this happens in the stroma because all of the enzymes that catalyze all of these steps, and there are bunches of them, they are dissolved in, or, dissolved in solution in the stroma. And I can identify the enzyme responsible for carbon fixation. Dun, dun, dun. That enzyme, let's get a different color, is Rubisco. Okay, Rubisco. Now, Rubisco does not end with ACE, which I said, you can say, Miss Pirro, you always said enzymes end with ACE. Well, Rubisco stands for ribulose bisphosphate or biphosphate carboxylase. So now you know why we abbreviate it Rubisco. So Rubisco is kind of like a nickname. So what it does is this ribulose bisphosphate is this. Carboxylase means to add a carbon-oxygen molecule, aka carbon dioxide. And so it is an enzyme that adds a carbon dioxide to RuBP. But we call it Rubisco for short because it's just easier. And that's it. So actually it wasn't as long as I thought. But that is all of the dark reactions, Calvin cycle, light independent reactions, all the same thing. And that is everything you need to know for those 10 learning targets. I hope that helps, but if not, ask me questions.